Today we have with us Dr. Yashraj Kulkarni. He's currently working as a postdoctoral researcher in the field of drug discovery at the University of Copenhagen. Truly an honor, sir.
Thank you for having me on the podcast. It's a pleasure to be here and share my experiences with you. And I look forward to answering your questions if you have them.
Welcome, sir. Can you please elaborate on your field of research as what you're doing right now?
Yeah, sure. So, as you mentioned, I'm currently working as a postdoc at Copenhagen in the field of drug discovery. Specifically, I work on drug design. So,
What do you mean?
I would just briefly mention the drug development or drug discovery process, which is where which is from the start to the end of developing a drug molecule. So it normally in today's timeline, it takes about 10 to 15 years to actually identify a target and start designing drugs on them. And then it's followed by clinical trials and regulations, and it takes about 15 years to come out in the market. So drug design happens in the initial part of this process, which is the first 3 to 4 years, where it starts with identifying what exactly in the body you want to target with your drug, and then you start designing molecules that target those drugs, those targets in the body, which are most probably proteins. So what I work on is designing peptides, which are short protein sequences, as drug molecules that can bind to certain targets. I specifically right now work on signaling proteins, specifically GPCRs, G protein coupled receptors. They are quite versatile, and they perform a lot of different functions. And they are a key target for pharmaceutical companies to design drugs on, because they regulate a lot of signaling processes inside the cells. So currently I'm working on developing methods to design new peptides that can bind to these GPCRs and can hopefully become drug molecules some point in the future. And I would also like to mention that I work predominantly with computational tools. There are people who work in the lab with chemicals. They run experiments, but I sit in front of my computer, and I actually model these biological systems on the computer. So I run simulations and I analyze data, and it complements with the results that we get from the labs. Entering into, I did my engineering in biotechnology, and that's when I realized that it's such a diverse field which has like at least 10 different subfields within it. But at the end of it, I was still not sure where exactly my preference was. So that's when I decided to do my master's, and I moved to Uppsala in Sweden, where I did my master's in applied biotechnology. So there, my intention was to dip my feet in different fields of biotech and sort of understand where I belong. So I worked with molecular biology, structural biology, industrial biotechnology, also protein engineering. So I did a lot of wet lab stuff. But then when I was working with structural biology, I was also introduced to the computational stuff, wherein I could view or visualize three-dimensional structures of protein molecules and other biomolecules. And that kind of fascinated me. I did my master's project also in that area, where I was actually simulating the movement or motion of these protein molecules. And then I decided to do a PhD in that field. So that kind of helped me navigate in a certain direction.